Hi, everyone. Thanks, Chris, and thanks to all the organizers. I really appreciate being here among all these great speakers. Today I'd like to talk to you a little bit about how we can scan for selective sweeps, and in particular, how we can do this actually using unphased data. Selective sweeps, beneficial mutations will leave these sort of very conspicuous signals in the genome, distortion of the site frequency spectrum, reduction of genetic diversity, and really long haplotypes. And there's a lot of tests out there that exploit these patterns to make inferences about where in the genome positive selection may have occurred. Today I'm really going to be focusing on statistics that look at the reduction of genetic diversity in long haplotypes. Just to be clear, I'm really talking about sweeps that are sort of ongoing or recently fixed. All right, just so we can orient ourselves, I have this little cartoon here of 10 haploid genomes. So the horizontal bars, gray bars, represent a chromosome, and the vertical colored bars represent different mutations and different alleles. So suppose we have an adaptive mutation that arises on this haplotype, and so this haplotype ends up sweeping through the population. So maybe it reaches 70% frequency in the population. And so assuming that this has happened somewhere in the genome, we want to know how can we capture this process in a statistic. So if you just kind of stare at this a little bit, maybe stare around the location surrounding, the genomic location surrounding this adaptive haplotype, you'll notice that there's a lot of low genetic diversity surrounding the adaptive allele. A lot of statistics, haplotype-based statistics, sort of try to capture this signal. And so essentially what you will do is you'll take a small window surrounding your test location, you'll calculate haplotype homozygosity, you'll make it a little bigger, you'll recalculate it, you make it a little bigger. And the idea here is that when you have an adaptive allele, this homozygosity will sort of slowly decay as you make that window bigger and bigger. But on the flip side, if there's no adaptation in this location, that homozygosity will decay quite quickly. And so there's a lot of statistics. IHS and NSL do this within a single population. They make these comparisons. Statistics like XP-nSL and XP-EHH will do this in one population with respect to some reference population. One issue with this is that these statistics require phased data. And the thing is, if you're going to phase your data, you either need pedigrees or long reads or a decent understanding of recombination in your organism. And so that's not always feasible. And so I'm going to show you, so based on Harris et al. In 2018, they showed that we can actually sort of collapse haplotypes into multi-locus genotypes. And actually, they showed that we can still retain some information about sweeps when we do this. So what do I mean here? So suppose we have a collection of phased haplotypes here. So the colored haplotypes represent each a sample of a diploid individual. And if we actually don't know what the phase is, we might just then collapse these into what I'm calling a multi-locus genotype. So a 0-0 homozygote gets represented as a 0, a 1-1 homozygote gets represented as a 2, and a heterozygotes get represented as a 1. And so we can collapse our genotypes in this way, and Harris et al. Showed us that we actually retain some information about sweeps still. So just to illustrate this with my little cartoon, so here's our sweep cartoon, and I'm just going to convert these into ones and zeros, and then I'm going to further then collapse these haplotypes into multi-locus genotypes. So now you can see actually, so in the original phased haplotype version, we can see there's a lot of low genetic diversity surrounding the adaptive allele. And actually, if you kind of squint at this a little bit, you can see there's actually still a lot of low diversity surrounding this adaptive allele. So actually, assuming that we have these multi-locus genotypes instead of phased haplotypes, we can actually sort of reformulate IHS, NHL, NSL, XPHH, and XP-nSL. And I'm just going to show you some results for these two, NSL and XP-nSL. They're generally the same for IHS and XPHH, but these look just ever so slightly better. To evaluate this reformulation, I'm going to simulate some sweeps with DisCol. So DisCol is a coalescent simulator, and it's currently the only one I can find that will let me do this efficiently in a two-population context. And then for this talk, I'm just going to talk about three different population scenarios. So one is a two-population divergence model where the two populations that we compare are the same size, one in which the reference population is smaller, and one in which the reference population is larger. And then for this talk, I'm really just going to assume that the divergence time between the two populations was 2,000 generations, that there's no migration between the two populations, and that the larger population is going to be an NE of 10,000, and the smaller population is going to be an NE of 5,000. And so first, I'm going to talk, I think, about NSL, and since NSL is a single population statistic, we're really just concerning ourselves with the scenario where NE is either 10,000 or 5,000. So, all right, so I'm going to present you this kind of complicated figure, so let's just walk through it a little bit. So on the y-axis, we have the power of the statistic that we can calculate, and then we actually simulate this across a number of different scenarios. So we're going to simulate a hard sweep where the adaptive allele is adaptive essentially from the beginning. And then we can also simulate a number of different soft sweep scenarios. So in this case, E equals 0.01 represents a scenario where the allele becomes adaptive at when it's already at 1% frequency. And then this goes 2%, 5%, 10% frequency. I also am going to simulate it across a bunch of different selection coefficients. And then on the x-axis here, this represents the Fs. On this side represent the frequency at time of sampling. So when we sample our genomes, the adaptive allele is at 70%, 80%, 90% frequency. We also have one where it's fixed. And then we have post-fixation sampling as well. So 50 generations post-fixation and 100 generations post-fixation. So hopefully now you can see this sort of massive tangle of lines here, but I think the take-home message here is that we're actually doing fairly well using unphased data, and we're doing pretty well in the sort of realm where we expect it to do well. And then these one population statistics are sort of really bad at identifying sweeps post-fixation. They're really good at finding them as they're sort of reaching relatively high frequency. And so, okay, I think the natural question you're probably all asking yourselves, oh, and this is for an N of 10,000. So you're probably wondering, okay, well, this looks okay maybe, but how does it compare to if we had, like, say, perfectly phased haplotypes? So on the right here, I'm going to compare now with a couple different contexts of the sort of having phase data. So so here on the y, I'm going to plot the power difference. So I'm going to take the difference, and basically up here the multi-locus genotype-based statistic is doing a little better. Down here, the haplotype-based statistic is doing better, and then this is still the same same parameters here on the x. And then I'm actually going to show you two different sets of lines. The blue lines are going to be where we compare the unphased statistic to the haplotype-based statistic just applied naively to unphased data. And then the red lines, I'm going to compare its performance when we have sort of perfectly phased data. So just pulling it right out of the simulation. Perfectly phased, we know we couldn't do any better. So when we look at this, actually we can see that we're doing sort of reasonably well in the one population case, over in the part of the parameter space where we expect to do reasonably well. So this first case, we actually seem to be doing like pretty decently. Although I'll note, like, if you do have perfectly phased data, you may as well use the phase statistic. So just to keep going with this, so now if we cut that population size in half, so now the sweep is happening in an any population of 5,000. We do start losing some more power. But except for all but the strongest sweeps, but we're still doing okay. And if we compare to the phase statistic, you can see that we're losing a little bit of power compared to the phase statistic, but it's doing all right. But now I'd like to. So this is NSL. I'd like to talk a little bit about using XP-nSL. So this now we use, we compare haplotype patterns in our query population, and we compare it to some other reference population, some sister population that we may have data for. And actually, so when the population sizes are equal and equal to 10,000, any of 10,000, we actually do like super well. Across pretty much all the parameters, including soft sweeps and even relatively weak selection. And then when we compare this to the phased statistic, even with perfectly phased data, we're actually doing more or less the same across the parameter set. So if we now look in a slightly different demographic scenario, so now the sweep is happening in our larger population, n equals 10,000, and our reference population happens to be a lot smaller, so n equals 5,000. We still do like pretty decent, especially right up to and including fixation. We start to lose a bit of power for the really soft sweeps and the fairly weak selection post fixation. But when we compare that to the perfectly phased or the phase statistic on perfectly phased data and on the naively on the phase data, we actually we're doing about the same, if not even sometimes a little better. And then when we look at then the opposite scenario, so supposing that our statistic, or sorry, our sweep happens in the small population, so n equals 5,000, and we're comparing it now to a sister population, sister population with n equals 10,000. Again, we get very similar patterns. So these XP statistics are actually doing like super well relative to having the phase data, and so I think we're actually in pretty good shape. So as an example, applying this, a couple of years ago, we published a scan for selection in rhesus macaques that are living at high altitude in China. So we had 23 samples living in regions with an average elevation over 4,000 meters, and 22 samples from a low altitude regions living sort of under 500 meters. And the standout signal here in this analysis was this little peak right here, which happened to land right in front of EGLN1. So probably most of you know EGLN1 is a regulator of oxygen homeostasis and is really just sort of a classic target of natural selection in organisms living at high altitude. So like I said, I used the phased statistic on this data, but I went back and I reapplied the unphased statistic, the unphased XP-nSL statistic. And so the original phased data is these red dots, and our unphased statistic popped out these blue dots, and actually doing like pretty well, and our sample size was not like super big. So first part of my talk, so this is my conclusions for this part. There's a lot of popular haplotype-based scans for selection out there, a lot of statistics, and actually these can actually be adapted relatively straightforwardly to use unphased data and still retain pretty good performance. I guess I'd have to strongly suggest, if you have the data, if you have a reference population that you can compare to, this clearly allows us to perform quite well. Maybe a little less good if you only have the single populations, and then I just want to point out, like, sample size here. One of the problems with this approach is your sample size is effectively cut in half, at least for diploids, and so that's something to be aware of. And then, like I said, the single population statistics, I think, they suffer a little bit of performance, but if it's the best you can do, then it's the best you can do. So this is the first part. And now I want to talk a little bit about going beyond summary statistics. So all these statistics that I just talked about are essentially summary statistics of the haplotype frequency spectrum, and they work pretty well to capture information and allow us to sort of identify putative sweeps. But I want to suggest that maybe we can sort of directly use the ranked haplotype frequency spectrum to make inferences. So by the ranked haplotype frequency spectrum, I just mean we look in some window and we look at all of our haplotypes and we rank them in order from most frequent to least frequent. So not only am I going to use the haplotype frequency spectrum, but I also want to ask a few questions. Maybe are we able to sort of infer other parameters of the sweep? Maybe the number of haplotypes sweeping, so it would be like softness, or the sweep width. So actually, I've been talking about haplotypes here, but what I really mean are multi-locus genotypes, because again, we're concerning ourselves with unphased data. But actually, so what I'm gonna present, all the theory and stuff, I'm gonna talk about it as if they're haplotypes, but it's actually essentially identical for multi-locus genotypes. So wherever you see haplotype, just replace it with multi-locus genotype. So, well, I guess I'm going to be surprised by what comes up too. Okay. So, all right. So, suppose we have a neutral region of the genome. It might look something like that. And so, if we go and we calculate our ranked haplotype frequency spectrum, we might expect to see something that looks like this. So, here's the most frequent haplotype, the second most frequent haplotype, etc. In the context of, say, a hard sweep, we have one haplotype sweeping towards fixation, and so we might expect then to see a huge distortion in this first haplotype here. So you have one haplotype that's at really high frequency, and then a bunch of others that are at lower frequencies. But that's a hard sweep. So what if we have a soft sweep? So maybe we have multiple haplotypes sweeping. In which case, say maybe if we have two haplotypes sweeping, we might expect to see the first two haplotypes having a distorted frequency, and then the rest down fairly low. Or even maybe we have four haplotypes sweeping, and so we might see this distortion of the frequency spectrum in the first four haplotypes. So I'm going to show a fair bit of math, but if that's not your jam, just sort of, you can blur your eyes a little bit and just listen to what I'm saying. What I'm going to suggest here is that we will represent our neutral or background haplotype frequency spectrum with some vector of numbers, and then we can represent our distorted haplotype frequency spectrum as a vector of these other numbers. And m here is a parameter suggesting that sort of m haplotypes are sweeping. So how do we actually model this distortion? Well, it's this horrible mess, and it's not particularly important that you try to understand what this is saying here, because I'm going to try and illustrate it up here. So what this is basically doing is we are drawing some line across the distribution here, the neutral distribution, and we're saying that if there's only one haplotype sweeping, we're just going to scoop up all of this mass on this part of the spectrum, and we're going to plop it onto the sweeping haplotype. And so that would be how we would model, say, this scenario right here. Similarly, if we propose that there's two sweeping haplotypes, we're going to scoop up all of this mass and distribute it across the top two. This little function determines how we do that. And then similarly with m equals 4, we scoop up this mass and plop it over here. Okay? So, all right. So this model was actually developed in 2020 by Harris and DiGiorgio, and they called it the likelihood-based approach for selective sweep inference, or LESI. So this was cool, and Mike and I started talking about it, and we decided that maybe we can do something a bit more. So actually, we started thinking about it, and we said, well, you know, at the site of a putative sweep, so suppose this little star here is where the sweep location is, so we get this distorted haplotype frequency spectrum. But if you look at adjacent windows, you know, adjacent windows should be in LD, at least if they're close, and so the frequency spectrum should look fairly similarly distorted. But if we move far enough away out, this haplotype frequency spectrum should start to decay, or should start to change towards what the background or the neutral distribution looks like. So we decided that we could actually model this. So we are going to represent a distorted haplotype frequency spectrum, a distance a away from the selected site. I'm going to represent it by this vector. And then we just take a very simple mixture model approach. So we take some mixture of the distorted model here, so what we expect over where the actual site is sweeping, plus the rest of it is some mix of the background. So when we do this, we calculate our mixture as just some decaying function of distance away from the putative sweep site, and we also simultaneously infer this A parameter, which is kind of related to sweep width. Okay. We really liked this method, and we were thinking about a name, and so we decided to call it the spatially aware likelihood test for improving LESI, or salty LESI. So how do we put this all together? So we can calculate a likelihood of observing this haplotype frequency spectrum under neutrality, and it's this. We can calculate then also the likelihood of observing the haplotype frequency spectrum under some model of distortion. This. And then we just can calculate a likelihood ratio test and maximize across these parameters, so we simultaneously infer M, which is supposed to be our number of sweeping haplotypes, A, which is our sweep width, and this epsilon is kind of just a nuisance parameter. Well, I guess I just explained this. So if we get an M equals 1, that would suggest a hard sweep. If we get an M greater than 1, this suggests a soft sweep. And then, as I said before, this parameter A is kind of, in a sense, related to the sweep width. So we did some simulations. So I believe this was a constant population size, again, of about 10,000, just for illustrative purposes. And each of these little plots here, so this new here represents the number of simulated sweeping haplotypes. So 1 sweeping haplotype, 2 sweeping haplotypes, 4 sweeping haplotypes. Our salty-lessy statistic here is the capital lambda, and then in parentheses we have a window size. So this is capital lambda, this is the statistic with a window of 201 SNPs, 101 SNPs, 51 SNPs, and then we compare against, then T is the original lessy statistic, H12 is a nice window-based statistic for detecting soft sweeps as well, and then we also put in NSL and IHS. So we can see generally that we actually do pretty well, even for soft sweeps. For sweeps that started, so we simulated this a little differently than when I presented before. On the x-axis, we have the number of generations ago that selection started. So this is actually all for the haplotype-based statistic. If we look at using this with MLGs, we actually can still do pretty well, but I just want to note that we need to reduce our window size in order to do pretty well. But you can see this dotted red line here is our salty-lessy statistic with a I'm running out of batteries here, is our salty-lessy statistic with a window size of 51. Okay, and then we compare actually, so I'll just point out this G123 is actually the, from Harris et al. 2018, this is the unphased version of the H12 statistic. So, okay, I'm going to apologize. Anyone who's been to the city knows that rats are not a small population, but I'm going to present this example anyway, so forgive me. But so we applied this, in the unphased case, we applied this to a data set of rats that were sampled in Manhattan. And so, you know, the idea here is that rats depend heavily on scent for communication, for behavior cues, and also, you know, finding food. And urban environments being both very harsh and noisy, but also very full of food. The paper where this data originally came from, you know, was really interested in actually looking for signals of adaptation in these rats that were living in this very harsh urban environment. So actually, we then just to demonstrate the use of the unphased version of this work, we took these 29 rat genomes, and this is the sort of Manhattan plot of their statistics. So higher salty-lessy statistic score, more evidence for selection, strictly speaking, more evidence for a very distorted haplotype frequency spectrum. And then we just chose a very strict cutoff using an outlier approach. So this bar, solid horizontal bar here, represents the top 0.1% of scores. So if we actually just look at where these two peaks fall, we can see that they fall on either olfactory receptor gene cluster or these calcium, what are they? Calcium receptor? Okay, they're actually expressed in the olfactory nerve layer in mice, and so if the same thing is true for rats, I think this is pretty sensible signal there. And so this hopefully is at least convincing that we're pulling out something that is of interest that we might expect. Just to wrap this up, we can use the haplotype frequency spectrum, or perhaps more relevant, the multi-locus genotype frequency spectrum, directly to use to scan for distortions that are consistent with selection. And actually, we can use these and actually retain pretty good performance. I didn't directly address this, so we can, in a sense, infer softness and width, but one of the things we found in our simulations is that this inference of softness and with width are somewhat confounded with both the age and the strength of the sweep. I think if you think about that for a second, that should probably make some sense. And then again, the same kind of caveat is that our sample size is effectively reduced in half for diploids, but in the case of the frequency spectrum, this is actually less of a problem. So I think with that, I just really want to thank you all for listening. My lab, both Annas are here, and they have some awesome posters. So please go talk with them. And thanks to all of you for listening.
Thank you for the wonderful talk. We do have time for questions.
Hi, thanks. That was definitely very interesting. I am not a theoretician at all, but I was just wondering, how do you decouple the effect of drift from selection? So I'm just assuming in a very small population, like say tigers or something, drift would fix some haplotypes or increase them. How do you decouple that?
So you have two options, neither of which are particularly appealing, but at some point when your population gets small enough, I think you're just kind of out of luck. So we typically will compare to some background, so the genome-wide distribution. So you can do that, and then the idea here is that the really, really extreme ones are sort of, hopefully those areas are enriched for true signals. But you run the risk that it might just be, a lot of drift that happened to really distort the frequency spectrum. The other option is if you do have a good idea of what your demographic history is, you can do a bunch of neutral simulations, and in our paper we do that for humans just to demonstrate an approach or how you might do that, and then you can use the neutral frequency spectrum that you simulate as your comparison in order to try and make that, but it's hard.
Thanks.
A quick question about your caveat about losing half your sample size. So in your simulations, though, when you know the truth, right? I guess your haplotypes there's no uncertainty. So is the trade-off in the empirical world that I can go half the size or do statistical phasing that may or may not be accurate?
Yeah.
And in that case, do you have thoughts on would the MLG approach be preferable?
Yeah, that's a good question. I mean, I think one of the things I still need to do is actually test this specifically with statistical phasing. So break up all the haplotypes and then statistically phase them to see, what actually the penalty or benefit would be. I did compare two perfectly phased data, and at least for the two population scenario, it works reasonably well. But yeah, it's a good subtle question that I need to work a little bit more to kind of suss out what the actual benefit or a detriment would be to choosing to statistically phase.
One more question. Annabelle, did you have one?
Mine was very similar, but I guess I can build off of it. Is just sort of, I was wondering whether for statistical phasing that many of us who work on models have, could it actually lead to not just a drop in power, but also actually like misleading results and false positives, and wondering if there was some way to kind of add that to why your method's a big improvement over that.
Yeah, that's I really appreciate all these questions, because I've been hearing that a few times now. So definitely, it's the next step for updating the preprint. We'll start looking at that a little bit. Yeah.
Super cool. Thank you.
Okay, let's thank Zach again.